Okay, Assalamualaikum. Previously, we have already looked at what is ATP, okay, what is adenosine triphosphate, and why ATP is used as the cell's energy currency. So today we are going to move to part 2 of chapter 12, and this time around, we are going to look at the actual process of respiration, of cellular respiration. Remember that the energy for respiration is obtained from the breakdown of glucose, and the energy released will be used to synthesize ATP, which is the energy currency in cells. Because, like what I have mentioned before, if all of the potential energy in glucose is released at once, then most of the energy may be lost as heat. So ATP is the best intermediary molecule to store this energy, since it can be broken down easily whenever energy is needed. So the breakdown of glucose occurs in 4 different stages. The first one is glycolysis. Glycolysis is basically the splitting or the lysis of 6 carbon sugar molecule, which is glucose, and it is broken down into 3 carbon sugar molecules, which is known as pyruvate. And glycolysis takes place in the cytoplasm of the cell. The diagram that you see here is the best way to explain the flow of glycolysis. Okay, in the first stage, glucose is phosphorylated, uh, because glucose is an energy-rich molecule. Right? Even though it's a rich energy molecule, it is also a very stable molecule, and it will not react easily. So to activate the glucose, okay, first of all, 2 ATP molecule is used to donate their phosphate to the glucose, and this will turn the glucose to fructose biphosphate, okay, which will later break down to 2 molecules of triose phosphate. So everything is shown here, right? So in order to produce energy in glycolysis, energy must be used first. Okay, uh, and then, okay, 2 hydrogen is then removed from both triose phosphate, right? You can see it here. Okay, 2 hydrogen is removed from both triose phosphate and will be transferred to 2 hydrogen carrier molecules known as NAD. Okay, so the NAD that you see here is what we call as a hydrogen carrier molecule because it carries hydrogen. Upon receiving the hydrogen, both of the NAD will be reduced, and this reduced NAD will be used in another stage of cellular respiration known as the oxidative phosphorylation, which we are going to look at a bit later. Uh, the end product of glycolysis is 2 molecules of pyruvate. Okay, you can see at the end of the flow, okay, you have 2 molecules of pyruvate here, and this molecule uh, actually still have a lot of chemical potential energy in them, right? Uh, those energy can only be harvested when free oxygen is available within the cell, right? And the process where those energy is harvested from pyruvate is known as the Krebs cycle and oxidative phosphorylation, which we are going to learn in a while. So the next stage in cellular respiration is the Krebs cycle, and in Krebs cycle, pyruvate that was made earlier by glycolysis will undergo further chemical reaction to release more energy in the form of ATP. But as you can see in this diagram, okay, in order for pyruvate to enter the Krebs cycle, it must first go through another stage known as the link reaction. Okay, this particular stage link the glycolysis, okay, link the product of glycolysis with the Krebs cycle. In link reaction, pyruvate will be decarboxylated and dehydrogenated. What is decarboxylation and dehydrogenation? Okay, it comes from the name, right? It is basically the removal of one molecule of CO2, that is for decarboxylation, and the removal of one hydrogen atom, which is dehydrogenation, from the pyruvate. Okay, the hydrogen that is removed will be transferred to another hydrogen carrier molecule, which is the NAD, which is going to reduce the NAD molecule itself. Right? So whenever the NAD receive hydrogen, okay, they will be reduced. Okay, and just like in glycolysis, this reduced NAD will be taken to the final stage of respiration, okay, to the final stage of cellular respiration, which is the oxidative phosphorylation, which we are going to look at a bit later. Once the decarboxylation and dehydrogenation have taken place, pyruvate will become a 2-carbon molecule known as acetyl. Okay, and this acetyl will immediately combine with a molecule known as coenzyme A, or in short, they like to call it as CoA. And the combination of acetyl with this CoA will produce a molecule known as acetyl-CoA. And the acetyl-CoA, okay, will combine with a 4-carbon molecule known as oxaloacetate to form a 6-carbon molecule known as citrate. Okay, and when this happens, CoA will be released. Only acetyl will actually combine with the oxaloacetate to produce the citrate, which is a 6-carbon molecule. The citrate is further decarboxylated and dehydrogenated in a series of steps to produce carbon dioxide, which is actually the waste gas that we breathe out during exhalation. And the dehydrogenation of citrate will also produce, reduce NAD and reduce FAD. And if you are wondering what FAD is actually very similar to NAD. Both of them are what we call as hydrogen carriers. Okay, so the function of NAD and FAD, okay, they take in hydrogen from other molecules and bring them from one place or from one reaction to another reaction. And that is why both NAD and FAD are called as hydrogen carriers. And at the end of the reaction, you'll see that oxaloacetate is going to be regenerated. Okay, and this regenerated oxaloacetate will be used to combine with another acetyl-CoA from another pyruvate. And this is why the whole thing is actually a cycle. And for each turn in the Krebs cycle, 2 carbon dioxide are produced. Okay, you can see it here. 1 FAD and 3 NAD molecules are reduced, and also 1 ATP molecule is produced. Okay, So I think that's pretty much it for Krebs cycle. Okay, you can actually see the whole thing uh, from the diagram. The whole flow is actually shown in the diagram. Okay, so the last stage in respiration is the oxidative phosphorylation, and this stage takes place in the inner membrane of mitochondrion. Okay, we call it as oxidative phosphorylation because both processes are involved in this particular stage. You have the oxidation process, and you also have the phosphorylation process where a phosphate is added to the ADP, okay, to form ATP molecule. Okay, and like I said, this particular stage takes place in the inner membrane of mitochondrion. Okay, remember that mitochondrion is an organelle that is made up of uh, two layers of membrane. So oxidative phosphorylation occurs in the inner membrane of the mitochondrion. In this stage, the energy for the phosphorylation of ADP to ATP comes from the activity of the electron transport chain. So what is electron transport chain? Basically, electron transport chain, as you can see in this diagram, is a series of protein carrier located on the inner membrane of mitochondrion that transport electron from one protein carrier to another protein carrier. Okay. Uh, Remember when we discussed about the glycolysis, link reaction, and also Krebs cycle? All of these processes produces their own reduced NAD and reduced FAD. So basically, all of those reduced NAD and reduced FAD produced will be brought to the electron transport chain, okay, for the final stage of respiration, which is the oxidative phosphorylation. Okay, at the electron transport chain, okay, the hydrogens are removed from both of the hydrogen carriers. They are removed from both of the NAD and FAD. And those hydrogen will then be split into its constituent, which is the hydrogen ions and the electrons. The high energy electron will be transferred to the electron carriers, right? Will be transferred to the series of electron carriers, and the electron carriers, the ETC, are actually arranged in such a way that when the electron are passed to them, the electron will move from higher energy level to a lower energy level. So as they go through this series of electron carriers, eventually energy will be released, right? Because they are moving from higher energy level to a lower energy level. Okay, so I repeat myself again. So when electrons are passed to the ETC and they move from one protein carrier to another protein carrier, energy will be released, and the energy that is released, if you look at this diagram, the energy that is released will be used to pump the hydrogen ions from the matrix of mitochondrion into the space between the inner membrane and the outer membrane of the mitochondrion, right? So this is the inner membrane, and you have one more here that is not actually drawn in the diagram, which is the outer membrane. So the hydrogen ions will be pumped into the intermembrane space, which is the space between the inner membrane and the outer membrane. Okay. So after a while, there will be a high accumulation of hydrogen ions in the intermembrane space, okay, compared to the matrix, right? Because as the electron is passed from one protein carrier to another protein carrier, energy will be released, right? And like I said just now, the release of energy will be used to pump those hydrogen ions from the matrix into the intermembrane space. Okay, so after a while, okay, you will end up with a very high concentration of hydrogen ions here. Okay, and once there is a very good build-up of the hydrogen ions, okay, they will eventually move back into the matrix through a specific protein channels at the end of the ETC. Okay, and the movement is down the concentration gradient, right? Because there are too many hydrogen built up in the intermembrane space. Okay, they will eventually move back to the matrix, but they will move back to the matrix through a specific protein channel at the end of the ETC. If you look at the protein channel that is located at the end of the ETC, okay, there is an enzyme attached to it, okay, which is the ATP synthase. So as the hydrogen ions move back into the matrix through the protein channel, the electrical potential energy released will be used to synthesize ATP from ADP. Okay, so the movement of hydrogen ions back to the matrix releases energy, and those energy is used by the ATP synthase, okay, to combine phosphate with ADP to produce the energy currency, which is the ATP. Okay, and this process is known. Okay, this specific process where hydrogen moves back into the matrix, releases energy, and the energy is going to be used to combine ADP with phosphate. Okay, this process is known as chemiosmosis. Okay, this process is known as chemiosmosis. And if you look at the diagram, you will see that oxygen, also plays an important role in the oxidative phosphorylation. Okay, as this oxygen become the final electron acceptor. Right? So the last structure or the last molecule that receive the electron are the oxygen. Okay, in the matrix of mitochondrion, oxygen will combine with the electron and also hydrogen ions to produce water. Okay, so that is the final reaction that takes place in the ETC. Right? The electron will eventually be received by oxygen. Where does the oxygen comes from? It comes from when you breathe, right? When you inhale, okay, oxygen will be absorbed by the cell, and those oxygen will be used as final acceptor to receive the electrons, okay, and combine with hydrogen ions to form water molecule. So that is basically how anaerobic respiration is carried out and how ATP is produced at the final stage of cellular respiration, which is the electron transport chain. So for a better understanding of the stages involved in the cellular respiration process, I'm going to share with you a few videos that shows all the 4 stages involved, right? Glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation on the electron transport chain. Okay.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
The Krebs cycle is the second step of cellular respiration. The Krebs cycle needs oxygen in order to proceed, and it takes place in the inner matrix of the mitochondria, which are found inside cells. If oxygen is present, pyruvate that is formed in the first step of cellular respiration is taken into the inner membrane and the Krebs cycle begins. During the preparatory stage of the Krebs cycle, pyruvate molecules left over from glycolysis give off a carbon atom in the form of CO2. Next, the Krebs cycle proceeds. The newly formed 2-carbon molecule combines with a molecule called coenzyme A. This produces a new product that is important to the Krebs cycle called acetyl-CoA. The reactions required for producing acetyl-CoA store an electron in a molecule called NADH that is used later during cellular respiration. The Krebs cycle begins when a 4-carbon molecule is combined with acetyl-CoA, forming a 6-carbon molecule called citric acid. Next, during a chain of reactions, 2 carbons are given off from citric acid in the form of CO2. 1 ATP molecule is produced. And since high-energy electrons are produced during this chain of reactions, 3 NADH molecules are produced. When NAD+ takes on an electron, it becomes NADH. So, 3 NADH molecules are produced. When 2 electrons are stored in a molecule called FAD during the Krebs cycle, it becomes FADH2. This chain of reactions produces 1 FADH2 molecule. We care about these stored electrons because they are used during the electron transport chain portion of cellular respiration, which occurs directly after the Krebs cycle. The Krebs cycle produces a lot of important molecules. When we started the Krebs cycle, we followed the story of one of the pyruvate molecules that was produced during glycolysis. But glycolysis actually produces 2 pyruvate molecules, so we'll have to double the amount of products produced by the Krebs cycle in order to have an accurate estimate of how much energy cellular respiration can produce from just 1 glucose molecule. The grand total for the Krebs cycle comes to 2 ATP, 6 NADH molecules, and 2 FADH2 molecules. The electron storing that has taken place during glycolysis and the Krebs cycle are used in the third and final stage of cellular respiration, the electron transport chain.
Most of the energy harvested from organic molecules during glycolysis and the citric acid cycle is stored in NADH and FADH2. These molecules give up their high-energy electrons in the third phase of cellular respiration, oxidative phosphorylation, where most of the cell's ATP fuel is produced.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondria. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
This illustration summarizes cellular respiration. Glycolysis in the citric acid cycle produce a small amount of ATP via substrate-level phosphorylation, but most of the cell's ATP is made via oxidative phosphorylation, when NADH and FADH2 produced in glycolysis and the citric acid cycle give up to oxygen the electrons obtained from organic molecules.
So the diagram that you see here summarizes the location of each stage in the cellular respiration. As you can see, glucose that is absorbed by the cell will be broken down to pyruvate in the cytoplasm. Pyruvate will then move into the matrix of mitochondria to undergo link reaction, where they are changed to acetyl and combined with a coenzyme A to form acetyl-CoA. Okay, acetyl-CoA will then move into the Krebs cycle, where it will combine with oxaloacetate to form citrate. And then all of the NAD and FAD produced in glycolysis, all of the reduced NAD and reduced FAD produced in glycolysis, link reaction and Krebs cycle will be taken to the electron transport chain, okay, on the inner membrane of mitochondrion for the final stage of cellular respiration, which is the oxidative phosphorylation. Okay, and then you have oxygen here. Okay, oxygen that is absorbed by from the blood vessels will be used as the final electron acceptor and will combine with hydrogen ions to form water molecule as a byproduct. So I guess we have covered all 4 stages in the respiration process. Please try to understand the flow of the process so that it will be easier for you to memorize all of the compounds and molecules involved. Okay, so I will stop here for today. In the next class, we will try to finish up chapter 12 because we only have a few subtopics left to be covered, including the anaerobic respiration. Please try to answer the questions that I have attached in the Google Classroom and submit it as soon as you can. And inshallah, I will see you in the next session. Okay, Assalamualaikum warahmatullahi wabarakatuh.